Welcome back to Biology 1110. In this video we are covering chapter 4. It's a smaller chapter, so we're covering it all in one video. And this chapter is all about carbon. Carbon is able to form a diversity of different bonds, and because it can form those different bonds, it is the base for a wide diversity of molecules. And the most important molecules of life are going to be based on carbon. So living organisms are based mostly of carbon-based, our molecules are based on carbon. And that is because carbon is it's unparalleled how good it is at forming large molecules, at forming complex molecules, at forming varied molecules. So all the big proteins, DNA, carbohydrates, these are the molecules that distinguish us as alive. These are all based on carbon. Organic chemistry is the study of compounds that have carbon in them. So when we're talking about a molecule being organic or not, we're not talking about farming techniques or pesticides or what kind of carrots you're buying at the grocery store. We're talking about molecules that are based on carbon and hydrogen. Organic molecules can range from simple molecules to colossally large ones. To be an organic compound, it must contain both carbon and hydrogen. It must have both of those. But organic compounds can also contain other atoms in them. It could have oxygen, nitrogen, sulfur, and phosphorus. Inorganic compounds might sometimes have carbon, or they might sometimes have hydrogen, but they won't have both. So water, which has hydrogen but not carbon, carbon dioxide, which has carbon but not hydrogen, these are inorganic compounds. We'll also see other inorganic compounds with sodium, calcium, iron, aluminum, and oxygen. Because carbon can form it has 4 valence electrons, so it can form 4 covalent bonds. So that means that carbon is tetravalent. It has a valence of 4. And because it has this tetravalence, instead of just being able to form one bond, being able to form 4 bonds, it can form these huge bonds. Typically we're going to see carbon having this tetrahedral shape. This is how those molecules and atoms are going to try to move themselves away from each other to give themselves as much space as possible so their electrons aren't bumping into each other. So here with methane is CH4. This is its ball and stick model, and this is how it's going to look in a space-filling model. Those hydrogens kind of trying to move away as far away in a 3-dimensional space as they can. Ethane, which is 2 carbons in it, and then hydrogens take up all the rest of those bonds. You can see this tetrahedral shape around this carbon, and this tetrahedral shape around this carbon. However, when carbon forms a double bond, like you see here in this ethene, it loses its tetrahedral shape because now it's bonded to 3 things. So it bonded to 3 things. It'll take this more of a planar shape, the same flat, where all the atoms are on the same plane. Okay. So this covalent compatibility of carbon is due to its electron configuration, and the most common things that carbon is going to bond with are hydrogen. You can see here, this is all these hydrogens. It also often bonds with oxygen and nitrogen in organic compounds. And these 4 atoms, carbon, hydrogen, oxygen, and nitrogen, is pretty much the main building blocks for living molecules. One other thing to mention on here, so ethane, you see this ETH prefix because we kept those same numbers of carbons the same, we kept that same prefix, but adding a double bond changed the ending. So you can actually tell what kind of molecule something is by the first part of it and also by the second part of it. We'll get some more into that, but that's just kind of an idea to hold on to for now. So carbon chains are going to be forming the skeletons of organic molecules, and because carbon can form these complex things, carbon chains can vary in length. So here we have ethane with 2 carbons, propane with 3 carbons. You could go on to build a carbon chain with 100 carbons. They can vary in branching. So here, this is not branched. This is branched. You could have another branch coming off here. You can have different branches as much as you want. Carbon chains can vary in double bond position. So here you have a double bond at the end of a molecule. Here you have a double bond in the middle of a molecule. That might not seem like a big deal, moving a bond, because you still have the same stuff, but that is going to change the shape of these molecules significantly, so that these molecules are going to react very differently. And also the presence of rings. So carbon can form a ring that is entirely made of single bonds, like you see here in cyclohexane. It can also have double bonds. Benzene is a common organic compound that has double bonds alternating with single bonds. 6 carbons. 6 carbons is going to be pretty much the most common size of a ring, although we can sometimes see rings made of 5 or 7 carbons. When we have these rings, we're not going to sit here and draw all of this out. So if you see a hexagon in something, that just indicates every point is a CH2, okay? CH2, CH2. Here in benzene, you can see it's alternating these rings to this shape indicates this molecule. So you don't have to draw out all your C's and hydrogens, just each point indicates where those are. So let's watch this video. I am not sure how this video is going to play for you when I'm making a recording. So if it doesn't work well for you, please go back, download the PowerPoint. It won't play from D2L, but if you download the PowerPoint, you should be able to get it to play for you.
The carbon backbones of organic molecules may vary in length and in location of single and double bonds. Molecules may be straight or branched and may even contain rings of atoms.
Okay, when we are talking about a molecule that is only carbon and hydrogen, that is a hydrocarbon. So these chains on here, these black dots are carbons, these gray dots are hydrogens. So this little tail is a hydrocarbon, this is a hydrocarbon tail, and this is a hydrocarbon tail. So many organic molecules like fats have hydrocarbon components. So although it has a head that has oxygen in it, these tails are hydrocarbons. Hydrocarbons are really good at storing a large amount of energy. So there's carbon and hydrogen bonds. The electrons there can be broken to release a lot of energy. Hydrocarbons are going to be named based on the number of carbon atoms that they have. So you've kind of already touched at this, but anytime you have one carbon, that carbon is going to be named, the one carbon molecule is going to have that prefix meth. So methane means one carbon with 4 hydrogens. The prefix ETH refers to something with 2 carbons in it. So ethane is 2 carbons and 6 hydrogens. Propane is 3 carbons and 8 hydrogens. And butane is 4 carbons and 10 hydrogens. And then besides differing in that, we can also differ, although we still have the same molecular formula. When we change the shape of those, we're going to be creating different molecules. So here, C5H12 is going to be pentane if it's in a chain, but if we start branching it, it gets a different shape. So this is going to be 2-methyl butane, so we're going to call it 4 plus 1 coming off. These are isomers. Isomers are molecules that have the same molecular formula, so these are both C5H12, but they have different structures, and because they have different structures, they're going to have different properties. They're going to fit into biological systems differently. Structural isomers are one type of isomers that they just have different covalent bonds. Their covalent bonds are shaped, they're rearranged in each other. Cis-trans isomers are a type of isomers that always have a double bond, but the stuff around the double bond is going to be in different shapes. So here we have 2 hydrogens and 2 X's that just stand for something special in there, right? So here those 2 X's are on the same side of the double bond. Here those 2 X's are on opposite sides of a double bond. So while a single bond can twist around and move, a double bond is fixed. So those are on this cis isomer, the X's are always going to be on the same side of the double bond. On the trans isomer, those X's are always going to be on opposite sides of those double bonds. And those X's are going to be shaped differently than the H's, so they're going to end up with molecules that are shaped differently. Then enantiomers are isomers that are mirror images of each other. And it's really easy to say they're just backwards from each other. They're pretty similar in shape. They must work the same, but they don't actually. Just like your left shoe and right shoe are mirror images, but you can't use them interchangeably. Your left shoe doesn't fit onto your right foot. It's the same thing with enantiomers. They don't fit into the same receptors the same way. They're not going to interact with your cells in the same way. Enantiomers can be important pharmaceutically. So if you have your two enantiomers, this can make the difference between like Sudafed and meth, like a decent cold medicine to make you feel better or a highly addictive illegal drug. The difference is they're isomers of each other. And ibuprofen, this enantiomer of ibuprofen is going to reduce pain and reduce inflammation. So you want this when you're feeling sick. This enantiomer is ineffective. So just by making the mirror image of it, it's not going to fit and it's not going to make you feel better. Albuterol is something that asthma patients will take typically through an inhaler to relax their bronchial muscles, to relax their airway, so that they're able to breathe when they're having an asthma attack. This R-albuterol is effective at doing all of that. The S-albuterol is not. And again, you can see they're just mirror images of each other. So organisms are sensitive to even subtle variations in molecules, that one enantiomer can be active and the other enantiomer be doing nothing. So let's watch this video about DOPA.
Isomers are compounds that have the same molecular formulas but different three-dimensional structures. The difference between isomers can literally mean the difference between life and death. L-DOPA, shown on the left, reduces the symptoms of Parkinson's disease. Its isomer R-DOPA on the right has no effect on the disease.
Okay. Then when we're talking about biological molecules, where you have molecules that are getting very big, and a lot of the importance of those molecules is going to be small little functional groups. So functional groups are going to be these little groups of molecules that can replace a hydrogen. So when you have a hydrocarbon, one of those hydrogens can come off and be replaced with a functional group. So if we are looking at these two different hormones, estradiol and testosterone, you can see they have the same backbone. Again, if you remember, we were talking about these are carbons at each point, and you fill in hydrogens. So it's got 3 rings plus this little pentose ring. They both have that base. They both have this methyl group and hydroxide group up here. But here, estradiol has a hydroxide group where this carbon on testosterone has a double bond to an O. And here, testosterone has a methyl group where here, estradiol does not. Those subtle differences are going to be what give estradiol and testosterone different hormonal groups to differentiate those lions into either a female or a male lion, which have very distinctive different properties as grown animals, just by these few little functional groups. So our functional groups are components of organic molecules that are most commonly involved in chemical reactions, and the number and arrangement of functional groups will give each molecule its unique properties. The functional groups we're going to talk about in class, you need to be able to, if I give you a name, you can tell me what its formula is. And then we're also going to talk about a couple of different properties of it. And on carbonyls, you also need to be able to differentiate them into these 2 different types of carbonyls. So we're going to talk about hydroxyls, carbonyls, carboxyls, aminos, sulfhydryls, phosphates, and methyl groups. Hydroxyls are an OH group. Sometimes it's written backwards as HO if you're dealing with the isomer of something. And these turn things into alcohols. So whenever you have an OH group, you have just made an alcohol. So here you took this ethane and turned it into ethanol. So ethanol is an alcoholic beverage, or an alcohol present in alcoholic beverages. So this is what converts grape juice into wine or hops into beer. It is going to be polar, so this will make, when this functional group is added to a molecule, it will make the molecule polar because that oxygen is electronegative, and that polar area will form hydrogen bonds with water. Next is a C double bonded to oxygen, so the C double bond O. That 2 lines indicates an equal sign. If the double bond is in the middle of a molecule, so you have a carbon double bonded to oxygen, and then the carbon is bonded to 2 other carbons, it's in the middle, these are ketones. And if the carbon is at the end of a molecule, so the carbon is double bonded to an oxygen, and it's got a carbon and a hydrogen, these are aldehydes. So make sure you can tell the difference between an acetone and an aldehydes. Sugars that have ketone groups are called ketoses, and sugars with aldehyde groups are called aldoses. And these are important in creating sugars. Carboxyl groups are found in organic acids, and they are a COOH. So it kind of takes those last 2 functional groups and combines them. You have a carbon double bonded to an O, and this carbon is also bonded to an OH. You are only ever going to see this at the end of a molecule because that only has one place left to be bonded to the carbon backbone for the rest of it. So these are going to act as acids. This hydrogen can reversibly be removed or accepted, and so these are going to give things the name carboxylic acids or organic acids. Here you can see this is vinegar, this is acetic acid. And because that hydrogen can be released, that's what these molecules will have a sour taste if there's something you eat. Sulfhydryl, so sulf, these have sulfur, and hydryl, these have hydrogen. So these are SH groups. These are commonly called thiols. So you have the rest of your molecule here, and then a sulfur and a hydrogen. So here is an amino acid that has a sulfur and a hydrogen on it. This is cysteine. One important thing is sulfhydryl groups can react. So you could have another amino acid over here with a sulfhydryl group, and those sulfurs will bond to each other instead of bonding to hydrogens. That will give a cross-link to different parts of a protein that can help stabilize that protein structure. And these are commonly called thiols again. Phosphate groups have a phosphate, and that phosphate is bonded to 4 oxygens. 1 oxygen has a double bond, then you have 2 oxygens with a single bond, another oxygen with a single bond that will go on and bond to the rest of the molecule. So phosphorus, double bond O, 2 O's, and then an O bonded to the rest of the stuff. These oxygens typically have a negative charge, and when these are attached to molecules, they will be able to react with water and release energy. The most common molecule that we'll talk about with a phosphate group is ATP, which is a nucleotide with 3 phosphate groups on it. And those phosphates hold a lot of energy in those bonds when you are digesting food and have a lot of extra energy, so it'll hold those energy. And then when you need energy to be released to do something, you can break those bonds and release energy to do the work of your cell. Next we have methyl groups. Methyl groups are just a CH3, which kind of looks like what you would expect on a hydrocarbon, but this is adding an extra carbon where you normally wouldn't. So here, this is cytosine. Cytosine is a base of DNA. And usually you'd expect to just have a hydrogen here, but instead you have this CH3, so it's methyl cytosine. And so you have modified this base of DNA. When you modify a base of DNA, you're going to affect the expression of genes. So this gene is going to be silenced. It's no longer able to take that gene and create RNA to create proteins. It can also affect the shape and function of sex hormones. We already talked about the difference in testosterone and estradiol having a methyl group on it. And these will give molecules the name of a methylated compound. So it's methylated DNA or methylated protein. Okay, so last of all, let's talk about ATP. I already mentioned that ATP is an important energy source in cells. In ATP, you have adenosine, which is one of our DNA bases. I hope that sounds familiar to you. Adenosine, and it's bound to 3 phosphate groups. So tri means 3, triphosphate, 3 phosphate groups. And these bonds between each phosphate group has a lot of energy. It can react with water and let one of those phosphate groups be released, leaving ADP, adenosine diphosphate. So adenosine has 2 phosphoruses on it and a heck of a lot of energy. And then ADP can break right along this bond and end up with AMP, adenosine monophosphate, and a little bit of energy. So whenever you need your cell to do some work, it will burn ATP. It will break it down and release that energy to do work. And when you eat something, you take the energy from your food and you can store it temporarily as sugar or fats and then break down those sugar and fats to create ATP. Okay, so that's it for chapter 5. It wasn't too much. You need to be very familiar with the types of enantiomers. You need to be very familiar with those types of functional groups. And you need to be very familiar with ATP. And those are kind of our big topics for this chapter. This is the only chapter. We'll probably start another chapter this week, but that is the main thing we're doing this week. So if you have any questions, please email me or contact me through D2L. You can talk to me during office hours or catch me after class. I hope you're having a great week. One more chapter and it'll be time for your first test. Bye.